Hello everyone and welcome to the very first webinar for Precision Analytical. My name is Mark Newman. I'm the president and founder of Precision Analytical. And today we have some exciting data and information to share with you about our latest innovation, which is the testing of organic acids, 6 of them actually, that we're adding into our Dutch Plus and our Dutch Complete profiles. So you'll be able to see those now on all of those panels. So those will be coming to you in your very next test. And this webinar is intended to give you some information to put those to use. So the 6 organic acids that we selected are 3 nutrient deficiency markers and 3 neurotransmitter metabolites. And as you'll see in this lecture, there are some fairly straightforward interpretation to those, whether they're low or high, but then there are also, in addition, some really interesting connections and touch points with the hormones and the metabolites that we're already looking at in our Dutch panels. So our medical director, Dr. Carrie Jones, is going to take you through that information and I think give you some really good tools to use in your practice as you continue to use the Dutch test in its now expanded form. But I just want to really briefly first just go through a little bit of just a couple slides of validation data, because it's really important that we establish for you that what we're doing is accurate and reproducible before we get into its clinical utility.
So you can see a little bit of data here, just showing at its most basic level, if we look at a liquid sample, if we look at our dried sample with the method that we're using, we're getting really strong correlation here. We've got one of our B12 deficiency marker, methylmalonic acid or methylmalonate. And then we've also got one of the neurotransmitters, the 5-hydroxyindole acetic acid or 5-hydroxyindole acetate, which is your serotonin metabolite. You can see further data here on the others that we are testing, but we do want to assure you that the values that we're looking at correlate to liquid samples. They're stable at room temperature for weeks, and the methods that we're using, which happens to be LC-MS-MS. There are a few options for organic acids, and I've developed these assays by several different methods, and we find that to work the best in terms of precision and accuracy. So it's really solid data, and the clinical part of it is even more interesting, and that's why we've invited our medical director, Dr. Carrie Jones, to come on with us today and to take a shallow dive and then a deep dive into the organic acids. So I'm going to hand it off to Dr. Jones.
Thanks, Mark. I am super excited to get this started. So let's get this started. I will be talking for about an hour. And so, and then of course, like Mark said, any questions you have, please put them in the Q&A. And at the end, we will cover all of them that we can. The questions we cannot cover just due to time, we will put in a FAQ sheet, a question sheet, and we will email that out to everybody. Along with the slides, you will get a copy of the slides and you will get a copy of the recording. We are recording this right now. Okay, so for those of you who have heard me speak before, you will know that I tend to cram a lot of information into my slides. And the reason I do this is I want the slides to be a reference for you. I want you to be able to go back to these slides and go, okay, these are the 10 things I need to consider, or these are the 4 treatments that I need to, really get behind and not have to flip slide after slide after slide trying to find the information. So today we're going to talk about all the organic acid tests we're looking at. So there are 6 of them. We're going to talk about what each OAT means, consideration for treatment. And naturally, we're going to talk about the relationship to the Dutch hormone results, your estrogen, your progesterone, your cortisol, your melatonin, and these organic acid markers. You're going to understand patterns, and we're going to do a case example at the end just to give you an idea of how it relates to the Dutch test and hormones and things of that nature. So for those of you I know who are listening that are brand new and don't know anything about the Dutch test, I do have 4 slides about us. Those of you who are already Dutch testers, first of all, thank you so much. Second of all, we totally love our practitioners. We have the best practitioners, we've decided. But just for those who are brand new, you have a lot of testing options. You have the Dutch test, You have serum testing, you have 24-hour urine testing, and you have traditional saliva testing where you spit in a tube and then get your hormones and your cortisol all out of those saliva tubes done throughout the day. So you can see here we have what Dutch offers compared to what other testing options offer, and what they're missing I have crossed out in red there. And while all the testing options are good definitely for certain things, it depends what you're looking for, and if you're trying to get a really comprehensive overview for your patient, for their hormones, for their HPO, HPG, HPA axis, and then adding on these Dutch extras like melatonin, 8-OHDG, and the organic acid tests we're going to talk about today, the Dutch really comes through, and some of the other markers or the other tests are missing some key points. So just keep that in mind. And if you don't know what you get with the Dutch, this is what you get. You get androgen metabolism, estrogen metabolism, progesterone metabolites. You get the metabolized and the free cortisol and cortisone. You do get the diurnal pattern, and you can get the cortisol awakening response. We do include that on our Dutch Plus. And then, of course, the Dutch extras. So the OATs, the melatonin, and the oxidative stress marker called 8-OHDG. The Dutch Complete is our flagship product. That's the primary dried urine test that you do 4 times during the day. Basically, your patient would urinate on these little strips of paper first thing on waking, 2 hours later, around dinner, and before bed. If insomnia is a problem for them, they wake up in the middle of the night, they can't sleep, then they would do a 5th strip that's included in the test kit in the middle of the night, and we do report the cortisol at that time. Now, our other big test is called the Dutch Plus. This is where we include saliva. Now, unlike the sort of traditional saliva companies that you're familiar with, this is where they actually use a swab, like a little cotton swab that they insert in their mouth and just get wet and then spit back into a tube. So there's no free form spitting that they have to do. They don't have to fill a tube halfway up with saliva. They just suck on these little cotton swabs on waking 30 minutes later and then 60 minutes later, and that collectively is known as the cortisol awakening response around dinner and before bed. On top of that, they do the 4 dried urine collections. So it's more collection, but more data points. And specifically with the Dutch Plus, we focus in the morning. So if you have patients that say, I have a lot of anxiety, I wake up with panic, I wake up when all my autoimmune symptoms are bad, I wake up and need a ton of coffee, then the Dutch Plus might be a good one for them. The Dutch Complete may be better for somebody who says, I can't sleep and I wake up in the middle of the night, or I just want a sort of more general overview of what my hormones and adrenals are doing. So what are the Dutch extras? On our test, we talk about them as organic acids and melatonin and 8-OHDG, but I want to show you this of what is an organic acid. Not many people know what an organic acid is, especially if you're pretty new to functional medicine or just new to sort of functional testing. So thank you to my friend Eric Balkevich, who, well, he doesn't actually even know that I swiped this from him, but I did swipe it from him. And so this gives the visual of how an organic acid forms. So if you look at the top part, if you're trying to get from A to D, then A requires enzymes and co-nutrients to get to B, and that requires an enzyme and co-nutrients to get to C, and then C also requires enzymes and co-nutrients. And of course, those enzymes are made by SNPs. So those of you who are into genetics know that genetics are involved here. But let's say A can't get to B. Let's say you have a genetic mutation and you can't get to B. Let's say you're missing a coenzyme, you're B6 deficient, so you can't get to B. What happens, that enzyme is missing because it doesn't have cofactors to make it in the first place. What if you have a hormone issue? What if you have a hormone issue that plays a direct role in the creation of that enzyme? You can't get to B. And so what happens is that A can go 2 ways. It can choose an alternate route. It can turn tail and go, all right, well, forget you, B, I'll go somewhere else. Or it gets excreted out into the urine as an organic acid. And so that is how an organic acid is formed. So we keep this in mind. We keep in mind the alternative pathways, which I'll talk about, and we'll keep in mind like trying to get to B, but it becomes the organic acid in the urine. So that's the definition of that. When you get your Dutch Complete or your Dutch Plus, this is what page 6 looks like. We call it the Dutch Extras. So it looks a little bit confusing, so we'll walk through each one. Especially if you're brand new to this or newer to functional medicine, newer to functional testing, where do you even start? We do include melatonin and the oxidative stress marker, 8-OHDG. I'm not going to talk about either of them in this lecture. That is a different lecture, but I will be focusing on the nutritional organic acids. Methylmalonate, MMA, which many of you are probably very, very familiar with. If it's elevated, it reflects an insufficiency in vitamin B12. Xanthuronate, if elevated, reflects an insufficiency in B6. And pyroglutamate, if it's high or low, it does reflect a problem with glutathione. Now, on top of that, we look at 3 neurotransmitter metabolites. We have HVA, which is a dopamine metabolite. And remember, it's not all about dopamine. It can reflect genetic mutations, nutritional issues, other neurotransmitter problems, adrenal problems. The same goes for VMA, which is a norepinephrine metabolite. And then there's 5-HIAA, which is a serotonin metabolite. And both VMA and 5-HIAA can also reflect nutritional, genetic, neurotransmitter, hormone, adrenal, what have you. Later in the slides, I will talk about certain foods that will affect them that you're going to want to be sure you avoid. And also be aware that these are affected, they're neurotransmitters, so they can be affected by things like SSRIs and SNRIs. And also, we talk about other sort of systemic stuff, for example, like the gut and serotonin and 5-HIAA. So in all our instructions, we do include, like things that will affect. So pay special attention to the instructions, but I do talk about it in these slides. So for those of you who are like, all right, got the basics. I know that already, but I want to really dig deep. I need the more advanced stuff. Here's where we go. Here's where we go through the rest of our, 40 some slides to get through each marker and what to do when they're high, what to do when they're low. So as my husband's best friend says, are you ready to go down that rabbit hole, Alice? Because here we go. Before we get started, please pay attention to words that are underlined. Unfortunately, the platform that we're using, Big Marker, italics didn't come through, but the underline did. So it says italics and underline. When you get the slide, I'll try to convert them back into italics. So if you print out your slides or watch them on your computer again, if you just pull them up separately from the replay, they'll be italicized. But what I wanted to show you was, if it has to do with estrogen or it has to do with cortisol, you can go look that up on the Dutch report and give you some more information. All right, MMA. If it's increased, it's a B12 problem. How is it a B12 problem? Well, they're either not eating it, they're not absorbing it in the stomach or small intestines, they're not getting into cells, or they have a genetic problem. So common symptoms of low B12, fatigue, poor memory, brain fog, unsteady gait, numbness, tingling, depression. These are just the most common symptoms. Obviously, there is a whole lot more, and many symptoms show up like this in your patient. I mean, how many of you have patients that come and list every single one off? Just make sure you don't forget about B12. Medications can interfere with B12, H2 blockers and PPIs for acids. How many people in this world are taking something because they get heartburn or they have, acid issues? Metformin or glucophage will affect B12. Birth control pills affect B12. Birth control pills affect just about everything, every B vitamin. Hydrocortisone and ciprofloxacin can affect B12. So just keep that in mind. How is methylmalonate formed? So here's my little graphical for those of you who are visual. So we have propionyl-CoA with the help of biotin makes methylmalonyl-CoA. And then methylmalonyl-CoA, you'll see the red circle around cobalamin, can either make succinyl-CoA, which goes to the Krebs cycle, or homocysteine and folic acid. Really, it should be folate. I swiped this out of a study, but we know it should be folate. Goes to methionine and then keeps going around the methionine cycle. So if you don't have B12, if you don't have cobalamin, then the arrow goes up to methylmalonic acid, or MMA. Now, methylmalonic acid and methylmalonate, or malonate, depending on how you say it, are the same thing. We have methylmalonate, but methylmalonic acid, it's the same thing. So if cobalamin goes down, MMA goes up. One other thing I'd like to point out here that Dr. Chris Masterjohn reminded me of, which is biotin. So but if you don't have biotin, you actually won't make methylmalonic acid or MMA in the first place. So something to keep that in mind. What if you have enough B12 and you can't just get it into your cells? How many times have you run a serum B12 on your patient and it was high, But you run an MMA as well, and it's also high, indicating there's a B12 problem. So this study showed there's a genetic mutation in the transport protein. So you have B12, but it can't actually transport into the cell, and now it's affecting B12 status. So for those of you who are into genetics, you may really, or who've done your genetic testing, you may want to really look at this and see, like, maybe you have a transporter problem as opposed to an absorption problem or some other issue. Oops, go back a slide. Getting ahead of myself. All right, MMA, how does it relate to the Dutch test? It is needed as part of the methionine cycle for the methylation of estrogen. Estrogen's underlined. Go back and check phase 1, phase 2, phase 1 going into phase 2 detox. Comped with SAM, of course. Comped is also responsible for the dopamine breakdown into HVA. So look at the HVA level. Norepinephrine into epinephrine uses the enzyme PNMT. That is a methyltransferase that also requires the methionine cycle to work well. And then norepinephrine and epinephrine both use COMT to break down into VMA. So there's lots of correlation here that go between your organic acid and your Dutch test. I also have an area called pseudo-Dutch related. Why are they pseudo? Well, obviously, because the whole body's connected. It's just one big system. So you do need B12 to break down protein and fats to amino acids and fatty acids. You need it, like I showed you, for succinyl-CoA for the Krebs cycle. That's, kind of important in the body. And you need it for methionine to make SAM, and SAM helps with methylation of myelin sheaths, phospholipids. So again, a lot of that brain stuff, the brain symptoms that show up with B12 deficiency. So here is another visual showing you that B12 is important as homocysteine swings around to methionine so that you can make the methyltransferase, which is circled there in red. So COMT or PNMT. So if you're doing genetic stuff, make sure you look at your methyltransferases and see what's going on, but B12 may be a key marker for you. Now, is B12 required? Not necessarily. So with permission, graciously, Dr. Ben Lynch let me use this slide, and wonderfully, I got to pick his brain somewhat about this as well. But remember, homocysteine can cut across to methionine with the BHMT enzyme, but it does need choline, it does need TMG, which is betaine, it does need zinc, and so there's a whole other subsection of stuff that has to be healthy and in the right levels for that little cut through to happen. Otherwise, you'll see on the left-hand side where methylcobalamin using MTR and MTRR, the enzymes made by those SNPs, then cycle homocysteine to methionine and around to the methyltransferase. Again, the methyltransferases are COMT, PNMT, really important for a lot of these Dutch markers. Now, people will say, should I use adenosylcobalamin or methylcobalamin? And Dr. Lynch reminded me, he was like, hey, if they have the MUT or MUT, a SNP issue, then methylcobalamin may not work for them, and you may have to just go ahead and give them adenosylcobalamin. Just skip the methyl altogether and go for adenylyl. And I was like, that's great for people to know, especially with Krebs cycle issues, especially getting succinyl-CoA to go into the Krebs cycle. So definitely keep that in mind when you're looking at genetics. So what do you do for B12? How do you help somebody with MMA? Well, it depends why their MMA is high and their B12 is low. If it's purely diet, you're talking to them about diet, they have a bad diet, or they don't eat enough, protein or B12 high foods, increase their liver, increase their sardines, which, let me tell you, are the two easiest foods to get people to eat, right? Lamb, wild-caught salmon, grass-fed beef, eggs, all those things are rich in B12. You can absolutely put them on B12 supplementation while you are trying to figure out why their B12 is low or what's going on. There are 4 options: methylcobalamin, adenosylcobalamin, hydroxycobalamin, and then, of course, cyanocobalamin. You can improve their HCl levels in their stomach. Why? Because stomach acid releases B12 bound to proteins in food. So if it's a stomach acid issue, a low stomach acid issue, but they're like, hey, you know what, doc, I totally eat grass-fed beef and I eat eggs and I eat liver, I'm one of those people, then make sure that their HCl levels are high enough to release the B12. You could consider testing for H. pylori. I know there are 2 camps on H. pylori. Some feel it is a pathogen and should be dealt with, and others do not feel that way. But if H. pylori is causing maybe problems and ulcers and issues, it's more out of control as opposed to under control, it can affect B12 absorption. Same for SIBO, maybe consider gut testing. Consider serum testing for intrinsic factor or parietal cell antibody testing, because that's how you suck your B12 down in. You need that intrinsic factor to be there to pull it through when you get it orally. You can consider biotin testing. Now, again, biotin has to do more when MMA is maybe blunted or low, but you still have all those same B12 symptoms. You're like, this is weird. Why do they have all these B12 symptoms? Really might be a biotin problem. So consider testing for biotin. And then those of you who are into genetics, I have the mutations listed there to help you decide which ones you might want to look at for B12 specifically. And again, as Dr. Lynch said, if you have the MUT mutation, they may need to take adenosylcobalamin as opposed to methylcobalamin, or just more adenosylcobalamin than methylcobalamin. All right, xanthuronate. If xanthuronate is high, your B6 is low. Now, B6 is a little more complicated than B12. It's either you're not eating it, you're not absorbing it in the small intestines, it's not getting activated to P5P or PLP in the liver. You have low cofactors, so you can't activate it, you can't get the tissue uptake, you can't get it out into circulation, or maybe you have a condition that affects your B6 directly, like pyroluria. So it is involved in over 100 enzymatic reactions. We all know B6 is critically important to so many things in our body, right? If the answer is not B6 or zinc, it's magnesium. So B6 just is one of those biggie vitamins that really does a lot for the body. Common symptoms you'll see, mouth issues, skin issues, blood sugar, insulin issues, fatigue, neuropathy, mental confusion, depression. It kind of looks like B12, right? And so the B vitamins are really pretty important for each other. Medications that interfere with B6, birth control pills, of course, anticonvulsant medications, and again, hydrocortisone and Cipro, the antibiotic Cipro, might affect B6. And so I know a lot of people, and this has been a really bad cold and flu season, and so a lot of people have been put on antibiotics, even though it seems, of course, most of the time it's viral. But if Cipro is something they're being put on, make sure you get their B vitamins back up. So how does it relate to the Dutch test? Well, a lot. So P5P that's the methyl form of B6. It is needed for methylation of estrogen. So check their phase 1, phase 2. P5P is important for the creation of neurotransmitters. So you may want to look at HVA, VMA, 5-HIAA. P5P is used in the transsulfuration pathway to make cysteine and thus glutathione. So check their pyroglutamate. It can complex with iron to increase 8-OHDG. That's a bad thing. We don't want high oxidative stress markers. We don't want 8-OHDG to be high. We don't want xanthuronate to complex with iron and cause all sorts of damage. So look at 8-OHDG if the xanthuronate is high as well. And then lastly, high xanthuronate, man, it can cause some real big problems for your blood sugar and your insulin. And there's some good studies to show this with diabetics. But of particular, gluconeogenesis needs B6. Glycogenolysis needs B6. And xanthuronate complexes with insulin. So now insulin is less effective to deal with your glucose. And now glucose is just running wild, causing all sorts of diabetes, pre-diabetes, blood sugar issues, because insulin's over here bound up to xanthuronate. So B6 can be really helpful at lowering xanthuronate and thus improving your insulin sensitivity and its effectiveness on glucose. Pseudo-Dutch related, things like excess estrogen, inflammation, and cortisol, which we test estrogen, we test cortisol, inflammation markers do obviously show up in your hormones. And then LPS stands for lipopolysaccharide from gram-negative bacteria in the gut, do push the kynurenine pathway away from serotonin. So if there's not enough B6 on this pathway, xanthuronate forms. So what you can do is address those things so you don't overload your kynurenine pathway, thus overloading your xanthuronate. And the kynurenine pathway does make NAD. So if it's going all the way down the pathway, NAD is important, but there are a couple other branches along the way of that pathway that might be actually damaging rather than helpful. And P5P does help enzymes get selenium from the dietary form. So if you're telling people to eat foods high in selenium for their thyroid, but they're B6 deficient, they're not going to get that benefit. So now they may have thyroid issues, is just one example. So again, for those who are visual, tryptophan can go down 2 pathways. So one, it goes up to the serotonin, and then that can make melatonin, or it can make 5-HIAA, which we test, both of those things. Only about 5% go that way. The other way it goes down is called the IDO, or the kynurenine pathway, and 95% goes that way hopefully to make NAD. And again, excess estrogen, excess cortisol, excess inflammation, and LPS from gram-negative gut bacteria push it the other way. So imagine if you have a slight drop in that 5%, you might have real big problems with your serotonin and thus your melatonin, but we'll talk about that with 5-HIAA. So again, just the visual of if you're new to the kynurenine pathway, you don't know where this falls, you can see tryptophan at the top, NAD at the bottom, and you can follow the arrows down and see where that goes. Or you can, look it up online, look up kynurenine pathway online and you'll see it. Now, again, I was showing the same cycle earlier when it had to do with B12, but look, homocysteine can go down the transsulfuration pathway to make cysteine. And if you don't have B6, you're not going to make cysteine. And then you're going to have a glutathione problem. So if you find that your glutathione is not being made, then you'd look to your B6. Maybe you've got a problem there as opposed to, everybody just giving cysteine for glutathione. How do you treat? So obviously you can increase B6 specific foods. So turkey breasts, grass-fed beef, pork, pinto beans, avocados, pistachios, chicken, sesame seeds, sunflower seeds, chickpeas. Chickpeas are really high in B6. You can supplement with B6 or P5P. Just be aware that high doses of pyridoxine can cause neuropathy symptoms. So low B6 can cause neuropathy, but if you overdose on B6, you can actually induce neuropathy. So you don't want to do that. That's bad. Nutrient testing for cofactors, riboflavin, B2, zinc, getting their mitochondria healthy so they make ATP, and then lysine. And then for those of you science nerds who want to know where all those cofactors go into, I wrote them all down for you because, of course, I had to know myself. But you'll see there, B2 activates B6 into P5P, into the active form. Zinc and ATP help with absorption, tissue uptake, and circulation from the intestines to the liver to the skeletal muscle. And lysine sits in the active binding site and acts as a Schiff base. And then it can bind and catalyze a reaction forward. So lysine can be actually quite important for that activation of B6. You can check the small intestines, do things like SIBO testing, or if you suspect they have leaky gut, they're eating foods that's causing inflammation. B6 is absorbed passively. It just sort of, ekes its way across. But if there's a lot of inflammation, intestinal upset, then it may just get pushed forward and not get absorbed at all. Of course, you can address the excess estrogen, the inflammation, the cortisol. You can address their LPS, and you can test them for pyroluria, which is a different test. But in pyroluria, those cryptopyroles bind up B6. So make sure you check their zinc status too, because zinc is pretty involved here as well. All right, pyroglutamate. Pyroglutamate is a glutathione problem. Glutathione is often indicated as GSH. So in some of my diagrams, you'll see GSH, and that's active glutathione. And if pyroglutamate is high or low, it indicates a glutathione problem. Either it can't be made, it can't be recycled, or it's being used up because you have all sorts of infections and things, oxidative stress, mycotoxin exposure that's sucking it dry. It does come from cysteine and glutamate and glycine, so look there. Common symptoms, obviously, it's a powerful, super powerful antioxidant in the body. And if you have problems with detoxification, if you're a chronically inflamed person, if you get sick or ill all the time, then maybe look to glutathione. Example, when this webinar is at prime flu season. Oh my gosh, it's like one of the worst flu seasons ever. And if people are telling me they're getting the flu 3 and 4 times, they think it's gone and it comes back, and glutathione might play a really big role there along with other antioxidants. Things that interfere with or decrease your glutathione, anything that affects your liver. Tylenol or acetaminophen, of course, goes right through the liver, and that can be a real big problem using up your glutathione. Any kind of environmental toxic exposure, solvents, chemicals, pesticides, herbicides, pollutants, cleaners, all that crap, heavy metals, EMF, alcohol, all that stuff, reduces your glutathione. It chews it up. And so just be really careful with what you put on your body and what you put in your mouth and what you have around you and what you clean your house with and where you buy your food and how much you drink. Like just really pay attention to that, especially if you have a glutathione issue or if you're chronically inflamed, if you're sick all the time, you have detox problems. That's the easiest place to start. So how does it relate to the Dutch test? Well, of course, it does help with estrogen detoxification. So unlike COMT, the COMT gene, glutathione is actually on a different pathway. It does help prevent the 4-OH or the 4-hydroxy from going down what's called the quinone pathway. So if anyone has ever heard me lecture about estrogen detox before, I talk a lot about this, where if 4-OH cannot get through methylation, it cannot go from 4-OH to 4-methoxy, then it turns tail, unfortunately, and goes down the quinone pathway, which commonly we call the bad pathway or the naughty pathway, because it forms adducts. We don't like adducts because adducts can cause DNA damage. And what happens is they bind to DNA and break off. And when you have broken off parts of your DNA, the body doesn't like that. And it's like, holy crap, we have holes. We have to fix it. And it sends in your DNA repair system. But if it can't repair as fast as the damage is happening, then you're increased risk for mutation. And now you're maybe increased risk for things like estrogen-based cancers. Well, what glutathione does, one of the big things is it helps excrete the 4-OH off the quinone pathway and helps, bind it up and get rid of it safely. And so for those of you with a lot of estrogen detox problems, male or female, check your pyroglutamate. You need B6, so check your xanthionate for the transsulfuration pathway. So check your pyroglutamate together with it. It does help handle infection and inflammation. And so for those of you who are familiar with Dutch testing, you know that there are really some similar markers that show up for those who are inflamed. For example, 5-alpha DHT, 5-alpha reductase can increase with inflammation. Estrogen can increase through aromatization. Inflammation can increase the aromatase enzyme. The 16-OH-E1, 16-hydroxy-E1 can increase. That's one of your phase 1 estrogen detox metabolites. Your 8-OH-DG can go up. Makes sense. You have a lot more oxidative stress. And of course, your cortisol may go up. Metabolized and or free cortisol can increase with inflammation, especially in active infection. So just be aware there. And then lastly, glutathione does recycle other antioxidants. It recycles CoQ10, which recycles vitamin E, which recycles vitamin C. And these are, as I just said, super potent antioxidants. And of course, they do a lot more in your body. So if you have glutathione problems, you may have other antioxidant problems. So how do you treat it? Well, obviously, first things first, address the cause. If you know they've had exposure, if you know that they have infection, you're working on their Lyme, you're working on their Epstein-Barr, you're working on their gut infection, you're reducing those things to reduce their inflammation, you're addressing their oxidative stress, that will help. It takes the pressure off. Remember, it comes from glycine, so glycine support, sulfate support, cysteine versus a lot of people will give NAC or N-acetylcysteine as a supplement, giving glutathione if it's appropriate. The cofactors are FAD, which is vitamin B2, selenium, making sure their mitochondria are healthy enough to make appropriate levels of ATP, magnesium. Mutation issues. So if you're interested in glutathione genetics, for sure, I have listed out some of the glutathione genes you can look into. And I'm talking a lot about genes in this lecture. And so for those of you who are interested, Dr. Lynch has his new book, Dirty Genes, that comes out, and he does specifically go into the glutathione genes as well as COMT. So that may give you some extra information, because I'm not touching on much more into genetics in this webinar, other than to say these genes, you need to look into. You can consider serum testing for GGT. Now, GGT is an enzyme, and it gets released when the liver is damaged. And so if your GGT is low or high, it may mean a glutathione problem. Why is that? Well, it's the start of the gamma-glutamyl cycle, the GGC cycle, and I'll show you that in the next slide. You want to ask them about their diet. Why? Because these are amino acids that build it up, and if they are not a protein-heavy diet, or not even heavy, just do they have protein in their diet? How many of your patients do you talk to and they're a carb and sugar-based diet? And that is not going to get you glutathione. It's not going to get you much of anything really, other than an expanding waistline, but it will not get you glutathione. And then lastly, you can test their methionine, and you can serum test their homocysteine and see maybe if methionine is high and the homocysteine is low, maybe they're not making cysteine, maybe that transsulfuration pathway is just not happening. All right, this was the picture that I was saying. You see GGT in the upper left corner, starting off that gamma-gluteal cycle. And then on the right-hand side is cysteine. And then as you follow the arrows down, 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 you can see where glutamate comes into play and ATP, and then you get to GSH. And again, GSH is glutathione. Now, glutathione has to get recycled, right? It gets used and recycled and used and recycled. And this is where selenium, you'll see Se in green. Selenium is on the left side. And FAD, which is B2, is on the right side. So If they're selenium deficient, which I talked about earlier, then not only will it affect their thyroid, it'll affect their glutathione recycling. So keep that in mind. All right, let's move into our brain and gut. And we call them neurotransmitter metabolites, but we know that obviously these can come up from other places, like 5-HIAA is your primary serotonin metabolite. But 90% of serotonin is made in your gut. Why? Well, serotonin, turns out, is helpful for gut motility, and it activates smooth muscle activity. So this may be important for the person with constipation. Maybe they don't have enough serotonin to get things moving. Or it's important for the person to get rid of foods that they, or bacterial strains that they ate and is causing food poisoning. So it will cause mass exodus very quickly to get it out of your body. So serotonin, it's not entirely understood in the gut, but we do know this stuff, and this can be quite helpful for people. Only about 1% is made in the brain, and the rest is made in the peripheral tissue. And believe it or not, it is stored in your platelets and your mast cells. Why? Well, it's serotonin in the periphery is released during injury and inflammation because of its analgesic, clotting, and vasoconstrictive effects. Super great if you get a cut, right? You get a cut, the platelets and the mast cells come in, they fix you right up, and your cut heals. Not super great if you have ongoing cardiovascular disease and smooth muscle vessel issues. And so now you have clotting and vasoconstrictive effect, and it can really be affecting cardiovascular stuff. So it's a balance, right? Serotonin's trying to help, but sometimes if you have perpetuated yourself along into a disease state, it can end up hurting you. So where does it come from? Well, it comes from tryptophan. And tryptophan needs biopterin, iron, and healthy estrogen levels to make 5-HTP. And what do I mean by healthy estrogen levels? Well, estrogen, when it's in the brain, it connects to estrogen receptor beta, it binds there, and estrogen receptor beta in the brain increases the mRNA to make tryptophan hydroxylase. And tryptophan hydroxylase is the rate limiting step for making 5-HTP. So if you have an estrogen problem, you may have a 5-HTP problem. And I'll talk about that a bit on the next slide. 5-HTP, that needs B6, so check your xanthionate, to make serotonin. And it uses copper and B2 with MAO to break down to 5-HIAA. Now, I said earlier I'm not going to go into melatonin, but I do want you to know what cofactors and genetics are involved with melatonin, because I get asked this quite a bit, and I'm going to go on a very teeny tiny little soapbox. But I oftentimes will suggest to somebody, like, you can give melatonin under certain conditions. And if I put it maybe out in social media, inevitably I get messages back from health practitioners who say, I can't believe you would ever recommend melatonin because of the rebound effect. And I'm like, okay, let me explain. Melatonin's rebound effect. If you go on melatonin and you go off of it and then you can't sleep again, you never address the cause in the first place. Why do you not have enough melatonin? Do you have a serotonin problem? Do you have a 5-HTP problem? Do you have an estrogen problem? Do you have a cortisol problem, an inflammation problem, an iron problem, a B6 problem? You see where I'm going with this? So practitioners get upset with me for recommending melatonin because they feel the rebound is a problem. And I'm like, well, why don't you just track back and find out where the problem is and address from there? Give melatonin as a Band-Aid in the meantime, get them sleeping, but then figure out where the cause is. Now, the other two things I need you to know are there are two genes to make melatonin. So there's AANAT and ASMT, and they require B5, which is pantothenic acid, and SAM, respectively. And I absolutely have had patients who've done their genetics and say, Carrie, I am homozygous AANAT. I'm like, well, you are going to have a problem making melatonin and probably need to be on melatonin because you're not going to make it. So in that case, I will put them on melatonin. I can give them all the 5-HTP in the world. I can give them all the B6 in the world. They're not going to make melatonin as healthy as somebody else. So if you have somebody that says to you, you should not use melatonin because of the rebound effect, you can have them listen to this part of my teeny tiny soapbox. Okay, 5-HIAA, if levels are low, address the cause. Just as I said, estrogen, cortisol, inflammation, all you can look for on the Dutch test. Check their gut if they have gut issues, because that will push them away from serotonin and down to the kynurenine pathway. But what happens if she has low estrogen? I just told you. her tryptophan hydroxylase can't make 5-HTP. So how many menopausal women or perimenopausal women do you have come into your office and they say, I'm heading into menopause and I feel more depressed? Why do I feel more depressed? Now you can say, well, unfortunately, you can't make tryptophan hydroxylase like you used to back in the day, and you can't make 5-HTP. Or women who are amenorrheic, they're skipping their periods, they have irregular cycles, big problem. It's also the reason the opposite occurs. They have PMS, and with PMS, they get depression. PMS is often due to excess estrogen, pushing them away from serotonin and down the kynurenine pathway. You just can't win with estrogen, can you? It's a balance. It's all a balance. Low cofactors, biopterin, iron, B6, copper, and B2. How many people come to you iron deficient, anemic, and depression is a symptom? Lots. So make sure you get their iron stores up or figure out why they're iron deficient. Of course, address the cause. Check genetics, MAO, check their diet. Tryptophan is an amino acid. It does come from protein. Please be aware, recent, even low levels of alcohol consumption can lower their 5-HIAA just because of the acetaldehyde dehydrogenase. So just be aware of that on testing. And low platelets and or mast cells can cause obviously low serotonin and then potentially low 5-HIAA. If they are on an SSRI or SNRI and their 5-HIAA is low, I don't think the medication is working. Just keep that in mind. The problem is likely tryptophan, not serotonin, but it may also be a serotonin receptor problem. But we definitely see this. So, check their diet, make sure they're getting enough tryptophan in the first place, or maybe work with them on getting their tryptophan up, or working with the practitioner who prescribed the SSRI to get their tryptophan up. High levels, well, they probably ate one of these foods. So butternuts are white walnuts, not butternut squash. So white walnuts, black walnuts, pecans, and their associated nut butters, plantains, bananas, avocado, pineapple, and pineapple juice, eggplant, and plums. If those are eaten the day before or the day of organic acid testing, they will raise 5-HIAA levels. So just be aware. Your patients will want to avoid these foods. So in the Dutch kits, now, for the last 2 months, we've had instructions in there that say to avoid these foods. If you are listening to this and you have older test kits, totally fine. They're still valid. Just remember to maybe print the slide off, give it to your patient and say, don't eat these foods the day before and the day of testing. If they are on 5-HTP, tryptophan, St. John's wort, SAMe, if they're on an SSRI, SNRI, tricyclic antidepressant medication, and their 5-HIAA is high, that's normal. It means it's working. So if you were hoping to get a baseline, they will have to go off their herbal and amino acid therapy for at least maybe a couple weeks, at least maybe 2 weeks. Please do not have somebody just stop their SSRI or SNRI or tricyclic antidepressant cold turkey. We do not recommend that even a little bit. Make sure they wean with their healthcare practitioner who put them on it. Or if you are capable of weaning them, it falls under your license scope, then you can wean them off of it. If they have malabsorption or gut issues, makes sense because a lot of serotonin is made in the gut. The most studied for serotonin is celiac. So if you're curious about those studies, you can PubMed search celiac and serotonin and see that. Stress will raise 5-HIAA, metabolic syndrome, like I was saying earlier with the cardiovascular disease in the arteries and the vasoconstrictive effect and the platelet effect. That's what they're linking it with. It is released from platelets due to injury and inflammation, as I said. They might have mast cell issues. This is very mixed in the research, but I know mast cell issues, histamine is hugely popular right now. And it's very complicated and it's hard. It's sometimes hard to understand. I even reached out to two super smart people, Elizma Lambert and Ben Lynch, and said, help me with mast cells. And they were both like, yeah, that's complicated. That's a whole other lecture. So I'm just going to leave it here as keep it in mind. And then lastly, carcinoid tumors of the enterochromaffin gland or cells, if the 5-HIAA levels are very elevated. So think gut tumor. All right, HVA, it's a primary central dopamine metabolite. It is made in the neurons in the brain, but in the periphery, it's made in small amounts in the adrenal medulla. There is no negative feedback when it comes to with HVA with dopamine. In the brain, we know it is part of the reward center. When you do things that make you feel good, you get that dopamine rush, and then of course you want to do it again. And that can be, quote unquote, healthy things like eating chocolate in moderation, or unhealthy things like people who do, cocaine or gambling or things like that. They get that rush and go back for more. Dopamine helps with motivation, prolactin suppression, sexual gratification, and motor control. Obviously, the most common motor control we know of is Parkinson's disease, with Michael J. Fox being the most famous person. But now lately, I don't know if you guys know this, but Neil Diamond was just diagnosed with Parkinson's. And so his dopamine is affected. So dopamine does not cross into or out of the blood-brain barrier. L-DOPA does. I'll show you the difference in just a sec. In the periphery from the adrenal, it does act locally, very locally in the tissue that it's in. It's a vasodilator to inhibit norepinephrine. It increases urine and sodium output. It reduces insulin output. It reduces GI motility, and it does reduce lymphocyte activity. So just an FYI that the dopamine coming from the adrenals does do those things. Where does it come from? It comes from tyrosine, whereas serotonin came from tryptophan. Dopamine comes from tyrosine. It uses biopterin and iron to make dopamine. Dopamine uses B6, B2, magnesium, and SAM to make HVA, or it's broken down then by COMT, and it can go on to make norepinephrine with copper, vitamin C, and the DBH enzyme. So you can see a lot of cofactors involved and similar genetics that you've been seeing all day, right? COMT and MAO. So a lot of history repeating itself. Now, VMA is your primary norepinephrine epinephrine metabolite. It is made in the brain and in the adrenal medulla, and it is stored. So cortisol is not stored, but norepinephrine and epinephrine are. They're stored in presynaptic vesicles. And when you get in a fight or flight situation, then they are dumped immediately. Microseconds are they released. But they should also be broken down fairly quickly. So the half-life of norepinephrine and epinephrine, just minutes, whereas cortisol's 1 to 3 hours, depending. So just a little history on that. There is no negative feedback here, and norepinephrine and epinephrine do not cross into or out of the brain. They should not be able to cross the blood-brain barrier. Now, the primary action is fight or flight. We know this. It's alert, attention, anxiety, vigilance, restlessness. It gears you up. It raises your blood pressure. It raises your blood sugar with glycogenolysis. It diverts things away from the GI tract out to the periphery, the skeletal muscle, because the point's to run. The problem is our fight or flight nowadays are not running things. We're usually sitting on our butt on our phone, and we get a stressful thing and we turn into fight or flight, but we're not running. So that's when the problems happen. Norepinephrine and epinephrine are supposed to be lowest in the night and highest in the morning. They somewhat follow a circadian rhythm, much like cortisol, unless they are stimulated. Now remember, they're stimulated within seconds because they are pre-stored in the synaptic vesicles. So if something wakes you up in the middle of the night, like for me, I was awoke at 4 a.m. to some weird sounds outside of my window. Turned out to be my neighbor who works the night shift, but he was not outside my window, literally. He was at his car in the street. But man, I woke up into a fight or flight, and all I could think about was my norepinephrine and epinephrine. And I thought, well, there go my low levels at night. They have just shot up. And not that long after, cortisol went up. All right, this is my most favorite quote that I have found from this research study. I just love it. I've posted it on social media because get this. Chronic stress, if continued for a long time, can damage many parts of the body. A significant part of damage is due to the effects of sustained norepinephrine release because of norepinephrine's general function of, ladies and gentlemen, pay attention to this, of directing resources away from maintenance, regeneration, and reproduction and towards systems that are required for active movement. How many patients come into you and say, in not so many words, they cannot maintain, regenerate, or reproduce? So many. Chronic stress. The consequences include sleeplessness, loss of libido, GI problems, impaired disease resistance, slower rates of injury healing, depression, increased vulnerability to addiction, all because of chronic stress and chronic norepinephrine release. Your body is trying to do what you're telling it to do. I'm in fight or flight. The problem is our fight or flight is not tigers and lions and bears, oh my, it's bills and family and stress and, not enough sleep and poor diet and the environment, oh my. So we get all these symptoms. For those of you who need a visual, phenylalanine uses biopterin to turn into tyrosine. Tyrosine turns into L-DOPA with biopterin and iron. That's converted into dopamine. Remember, dopamine doesn't cross into the blood-brain barrier. L-DOPA does. Dopamine can turn on into norepinephrine with copper and vitamin C or become HVA. And norepinephrine can turn into epinephrine with cortisol, SAM, and PNMT enzyme from the PNMT SNP. Or it can turn tail and become VMA if those things aren't available. Genetics. If you're a genetic guru or visual person or wanting to learn more, here's where the genes line up. And you can see where dopamine, norepinephrine, and epinephrine and the associated genes that make these enzymes that make these things happen. Please check to see if they're on tyrosine or DLPA. DLPA stands for D-L-phenylalanine. Dopamine or Mucuna supplements. Now, practitioners, how many times have you had a patient come in on something that you didn't put them on? So if you're consulting with Dutch and their VMA and HVA are high, and we say, are they on any of these supplements? Tyrosine, DLPA, dopamine, what have you. And they say, and you're like, no, I didn't put them on any of those things. Remember that patients will put themselves on things. They have other practitioners in their life. And so that can be a real problem. Check their supplements. If they're on SNRI medication or tricyclic antidepressants, if they're on uppers, if they're on amphetamines that are bad, amphetamines that are prescribed like ADD medications, if they're on appetite suppressants like phentermine, anything that's caffeinated pills or drinks, anything with epinephrine in it like Sudafed, which is either prescription or over the counter, depending on what state you're in the United States, that will cause it. If they're on opioid pain medications, their HVA tends to go up. Their VMA may be normal, but it may be elevated. If they have elevated adrenal output, you may get some crossover, some HVA VMA issues, and so check their cortisol. If they're an inflamed person, then because inflammatory cytokines cause increased sympathetic output. And then lastly, and of course, most concerningly, if they have a neuroblastic tumor or if they have pheochromocytoma, which is an adrenal medulla tumor. So if the levels are high and they've, and you can't figure out any reasons why they would be high, none of the other crazy symptoms, or they have, excuse me, I take it back. If their levels are high and they have really crazy kind of ramped up anxiety, sweating, high blood pressure symptoms, check them for a tumor, and we'll try to help you with that. Now, if HVA is high but VMA is low, so HVA, dopamine is not converting into norepinephrine. First things first, check their copper. Do a serum copper on them and check their vitamin C. Or we know humans can't make their own vitamin C. So if they have adrenal problems, if they get sick all the time, just put them on vitamin C, right? Just put them on vitamin C and check their serum copper. And now you can help their DBH enzyme move dopamine forward into norepinephrine, and the VMA will restore itself. If they come to you and they say, I have all sorts of gut issues, time to poop in a cup for science, because you need to check them for C. diff, Clostridial difficile, and see if that's the reason. If they ate bananas the day before or the day of testing, that will affect it. If they're on quercetin supplements, that will affect it. Check their adrenals, of course. If they have low levels of cortisol, then maybe their VMA is low because the adrenal gland, the HPA axis in general, is suppressed. And so the medulla is not putting out norepinephrine, epinephrine, and you're not getting cortisol output either. THC use. Now, the studies on THC, THC is hard. And I mean THC, not CBD. I mean THC. If the reason it's hard is they'll do short-term, they'll do long-term. It is long-term greater than 2 weeks, is long-term greater, if you ask somebody how long THC long-term is, most people would say a long time, right? Like 10 years. Oh, I've been using marijuana for a long, long time, as opposed to someone who's just done it in the last week. But they say short-term use, which they didn't really quantify short-term, might raise HVA. So just be aware. Opioid pain medication. And then a rare thing is called a DBH enzyme deficiency. They often might look like POTS, orthostatic hypotension, exercise intolerance, ptosis of the eyelid, psychological or behavior disorders. Typically, this is picked up early childhood, but if the enzyme is only kind of broken or kind of deficient, not fully deficient, then it may manifest worse and worse and worse into adulthood. And lastly is a neuroblastic tumor. Now, if HVA is low and VMA is high, obviously check the adrenals. If they have high cortisol levels, then most likely they're going to have high norepi as well, most likely, and they're going to have high VMA. If HVA may be normal and VMA be elevated with all the stimulatory stuff again. So if they say to you, I'm on ADD medication, or I'm taking phentermine for weight loss, I drink heap loads of caffeine, or I do caffeine pills before I work out. It's in my pre-workout, or it's the cold and flu season and I get sinus infections, so I'm on Sudafed. That could be a reason. If they're on especially risperidone, which is a dopamine reuptake inhibitor, and then of course, pheochromocytoma, which is the adrenal medulla tumor. Number 5, I have written there, higher risk for myocardial injury and dysfunction with elevated cardiac troponin 1. This was interesting in the research. Higher VMA and high elevated levels of cardiac troponin 1 it definitely means higher risk for myocardial injury or dysfunction. Now, do you run out and have everybody tested for elevated cardiac troponin 1? No. Consider their age, and please consider their cardiovascular disease risk. If you have somebody who's in their 20s, totally healthy, no cardiovascular issues, then don't worry about it. And if you have somebody maybe who's quite a bit older and has a lot of cardiovascular disease issues, you may want to worry about it. So I have it listed. All right, lastly, if HVA is low and VMA is low, look at the cofactors. Biopterin, are they iron anemic? Do they have SAM? Is their methionine cycle working? Do they have magnesium, copper? Do a serum copper on them? B2, NAD, are they not eating these things? Are they not absorbing these things? Do they have gut issues? Check their genetics. Do they have COMT issues or MAO issues that are really slow? And so you can't make HVA and VMA because the genetics have slowed the enzymes way down. Check their diet. If they don't have a protein-rich diet or they don't absorb their protein, then they're not going to get tyrosine, the amino acid tyrosine. Check their adrenals. If their HPA axis is totally depleted, then you're probably going to have depletion when these markers as well. Check their oxidative stress. PEA is phenylalanine. So if phenylalanine might be going down other pathways, look at their 8-OHDG. Do they have fungal or candida infections? You can get fungal and candida infections in lots of places. Do they have it in their sinuses, in their mouth, in their gut, in their vagina? Like really, ask them about this sort of stuff. This will affect their tyrosinase activity and they can't make L-DOPA. Glyphosate exposure. Glyphosate will block the shikimate pathway, and now they don't have tyrosine or tryptophan. So you'll often see low HVA, low VMA, and low 5-HIAA. And I don't know if you guys know this for sure. You can PubMed it, but in acute active glyphosate exposure, Roundup exposure, did you know that apple cider vinegar, like the real apple cider vinegar with the mother, has the little bacteria in it that can eat up glyphosate? Now, if you've been exposed and you're storing your glyphosate, it can't reach that. But if you've got acute exposure, then get your apple cider vinegar out and protect yourself. And then lastly, THC, again, long-term use maybe is quite suppressive. More research needs to be done and to sort of quantify like what is considered long-term use. Everybody has a different opinion of long-term THC use, don't they? All right, lastly is a quick little case presentation and we're done. So, to the questions, of course. So depression and anxiety on a woman with a SSRI. So she's a 50-year-old female. She came to the clinic. She was on an SSRI, but she said, I'm still depressed. I still have anxiety. I'm tired. I have irregular cycles. Makes sense. She's 50. And at the time, she was not reporting hot flashes or night sweats. So these are what her organic acids look like. But let's narrow it in so it's not so overwhelming and confusing. Boop, there you go. So her HVA is high. And of course, if it's a dopamine problem, that can cause anxiety. Her VMA is low. So if it's a norepinephrine problem, it's fatigue. And her 5-HIAA is low. And if it's serotonin, then that can cause depression. Obviously, there's a lot of other issues involved, cofactors, hormones, things like that. I get that, genetics, but this is just the basics. Dopamine, norepinephrine, epinephrine, serotonin. All right, so what would you do for this woman? Well, high HVA. First, I would say, let's check your copper, because to get from dopamine to norepinephrine, you need copper and vitamin C. She's tired, she's 50. I'm guessing, let's be honest, in this day and age, most people are probably stressed out. I could test her vitamin C or just put her on some vitamin C. I'm going to ask her about her gut issues. Hey, maybe we should do stool testing. If you've been on antibiotics recently, if you've got C. diff, because C. diff will stop the dopamine to norepinephrine conversion. Why is her VMA low? Well, I'm going to address the top things, and I'm going to look on the Dutch test at her adrenals to see if the whole HPA axis might be downregulated. And why is her low, or 5-HIAA low? It doesn't appear her SSRI is working, especially as she's still depressed. So I said, I think it's a tryptophan issue, and you can't make serotonin, and your SSRI is not working. On top of that, she's near menopause. She has irregular cycles, and she's 50 years old. So I know sometimes, some months, she has very low postmenopausal ranges of estrogen. And we know estrogen is needed to stimulate tryptophan dehydroxylase to make 5-HTP, to make serotonin. Again, remember, menopausal women, common complaint is depression, low mood. It's because of that estrogen. And then lastly, I'm going to talk to her about her diet, because maybe she doesn't have enough tryptophan because she doesn't have enough protein in her diet. And I'm going to talk to her about alcohol, because alcohol will cause low levels of 5-HIAA. So if she drank the night of testing or the day before testing, I'm going to ask her about that. And so that's how you would, that's just a simplified version of how you would take the 3 markers on the Dutch test, a female patient with estrogen issues, and where I would look. Boom, 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 just like that. So having the most comprehensive, easy to collect adrenal and hormone test available now with Dutch extras, including organic acid tests, allows you to make a greater impact on your patients at a much deeper level. So I do have 2 pages of references and resources. In particular, I would definitely personally like to thank Richard Lord and Ben Lynch, Chris Masterjohn, Elizabeth Lambert, Eric Balkavage, Michael McAvoy, and Sean Bean, who every time I ask for organic acid help, were quick to give me so much information, images, resources, studies. I really appreciate it. And you can see on MTHFR support, they have which B12 is the right one to use. And then I have a number of those research studies listed here if I didn't already list them in the slides. So you can print them out and look them up for yourself. So thank you so much. I really appreciate it. I told you I'd talk for about an hour, and I talked for literally about an hour. I pack a lot of information in. I know I talk fast. It's a good thing this is recorded, and you can listen to it again and again and again. We will start doing the questions. We have a lot of questions. So let's move on to the questions. And I think Mark is going to take it away from here.
All right. So that was terrific. Thank you, Dr. Jones. We've got some good questions. Thanks to everyone for supplying those. Just to remind you, some of you have really specific questions, still awesome questions, and we will respond to some of those over email, or we may even send them out, the answers to the whole group, but we may not be able to hit up all of those today, but just a few logistical questions that people have had. We've added 6 organic acids, so is the price changing? The price of the Dutch Complete and the Dutch Plus remains the same. So as we've increased efficiencies over the years, our approach has been to reinvest in the test. So initially we added melatonin, no price change. Then we added 8-hydroxydeoxyguanosine, no price change. And now we're adding the 6 organic acids. Same approach, the price is the same, you're just getting more information. Do you offer just the oats as a standalone panel? So we will have very soon, not the oats as a standalone panel, but the Dutch extras. So you'll get the oats plus the melatonin and the 8-hydroxydeoxyguanosine by itself, and that'll be a separate panel. Do you plan, this is a question I've got a lot, do you plan on expanding? Some of these organic acid panels are, $300 or whatever panels, and they include a whole lot of other stuff. We do plan to explore that, so I'm not sure what direction that will go. Some of it makes a lot of sense. Dr. Jones mentioned, hey, if you're biotin deficient, then that's going to change the interpretation of some of this. Well, there just so happens to be an organic acid, beta-hydroxyisovaleric acid. That one we've actually already validated. So we have some tests that we've actually already validated, and we're working through whether the clinical validity fits in with what we're doing. There's some other organic acids that we're not entirely sold on yet in terms of their clinical relevance. So stay tuned for that, but definitely something we've been exploring. So in terms of food, so Dr. Jones mentioned if you eat some foods, it can increase levels. We don't want that, right? We don't want serotonin to go into your gut from your food and then get turned into these metabolites. That doesn't really reflect your systemic levels of serotonin. So we do want you off the foods for 2 days. Some of the supplements, same thing. And if you have old kits, she mentioned taking a picture of her slide. That's an option. We also have inserts into our new kits that you can call, and we can give you a link to just print those off and say, look, here's your kit. You need this as well. Please follow the food restriction lists on here, and that will be helpful to get a more accurate look at the organic acids. Now, what if they're on an antidepressant? Now, that is where we get into this situation where it depends on what you want. If you're on an SSRI, what does it do? It makes your serotonin hang around longer, right? So if your serotonin's hanging around longer, what you should see is an increased level of serotonin, right? And so you'll get a higher level. That's a true level of increased serotonin. And Lisa, good question. So if you're on an SSRI and you can't make serotonin in the first place, then it's not going to work. That's right. And what you'll see is I can think of a particular example where somebody was on an SSRI, they were still depressed, and their metabolite it wasn't that it just wasn't high, it was actually low, which tells you that upstream from serotonin, there's likely a gross deficiency, right? So that's something you need to think through. If you want to get a baseline level of what does my patient look like without the influence of an SSRI, then yeah, you're going to want to get off of that for, I honestly am not sure how long that would take, probably at least a couple weeks of being off of the influence of the SSRI to see what their baseline levels are for that particular metabolite. If you're on 5-HTP, it's a different situation. Now I'm eating something that's in that pathway, which means you're going to get very high levels of the metabolite. It's not that relevant, right? So I'm not going to tell you get off your 5-HTP, but I will tell you if you are on 5-HTP, the levels of the serotonin metabolite are not relevant. They'll be very high, and that will be largely from gut clearance of that. Lastly, I may go back on the slides, or maybe I'll just talk my way through. I did see a number of questions about this kynurenine pathway. Super interesting, but a little bit confusing. So well, I'll just talk through it. So if you are looking at the kynurenine pathway, what happens? Tryptophan has two options, right? Down the kynurenine pathway, which is most of it, 90+%, and then the serotonin pathway. So cortisol, estrogen, and inflammation push tryptophan away from serotonin and towards the dominant pathway. So the dominant pathway gets even more dominant when you have high cortisol. So this is really interesting, is part of the mechanism of how stress and high cortisol can, quote, cause depression, is that your tryptophan pushes towards the kynurenine pathway, and now there's less substrate for your body to make the serotonin that you need. So the kynurenine pathway goes towards serotonin and away from serotonin, and it's pushed away from serotonin by cortisol, estrogen, or inflammation. Now, when it goes down that kynurenine pathway, it needs B6 to get to the end. If it doesn't, if there is insufficient B6, it makes a left-hand turn towards what? Xanthurenic acid, right? So xanthurenate is part of the kynurenine pathway, but you only get high levels of it if one of the doors is closed. So the door is B6. If it's not there, then you're going to make a lot of xanthurenic acid. And this is where this stuff gets really interesting, as Dr. Jones mentioned, that xanthurenic acid is not only an indicator of needing B6, but it itself is actually bad for you because it complexes with insulin, and on the story goes. So some really interesting tie-ins there, and this is why we added these markers to the Dutch test, is because it ties in with estrogen and cortisol and things like that. I have one more question for me, and I'll turn it over for some questions we've had for Dr. Jones. Should my patient stay on their hormones or vitamins when doing the test? So your hormones, your vitamins, B12, B6, they don't interfere with the test. So you just have to make a decision of whether you want to see your patient while they're influenced by these vitamins or not. And if you don't, and you want to see more of a true baseline, then you need to get them off of them. The only thing that, quote, interferes with the test is when you take something that feeds right into that pathway, like 5-HTP. So with that, I am going to transition over to some of the clinical questions that we've got and allow Dr. Jones to field those.
Thanks, Mark. First, I think the most common question that we've gotten through the entire webinar is, are we going to give the slides? Yes, absolutely, 100%. You will have all of my slides and Mark's slides. Yes, you will get those. The other thing I want to say too, for those of you who are still on and still listening, thanks. But we also want to know, we want to hear from you too. So please give us feedback and please let us know how we did. You can see it info@dutchtest.com and what other webinars you want to learn about. This was only an hour. We have so many questions that are so fantastic, but we can't get to all of them in an hour. So we're going to have to do a whole lot more webinars. Stay tuned. All right, question, I got one. Why I mentioned 4 forms of B12. Which one does my patient need? I don't know. That's why it's called individualized medicine. The most common, of course, are adenosine, methoxy, and hydroxy. It does depend somewhat on their genetics, of course, and some other markers. And so you might have to do some digging, or I know some practitioners just start with methyl. And if that's not working, as they're waiting for the genetics to come back or what have you, then they'll move on to adenyl or adenosine or hydroxy from there. Number 2, my patient already eats really good diet, a lot of the foods you already mentioned. Why wouldn't their nutrient markers be better than they are? Unfortunately, I think this is a byproduct of our food industry and our depleted soils and the choices that a lot of sort of standard American diet stuff. And I know that there are a lot of international listeners, and you understand what I mean when I say standard American diet, unfortunately. And I think that's why. Now, obviously, there are people who eat extremely healthy. They eat local. They eat, free range and organic. And even still, it just calls to our depleted soil and all the environmental stuff. And I think that's part of the problem. But it also might be absorption, right? Maybe they are eating perfect food in perfect amounts, and look at their gut, look at their stomach, go there. Because it's not you are what you eat, it's you are what you assimilate, right? So if you can't assimilate, that's the problem. When should I retest the organic acids, or maybe even when should I retest the Dutch test? That is also rather individualized. If you have somebody who is not well and you are having to make a lot of adjustments and work slowly with them, you may find you want to retest them at the 3 or 4 month mark. If you have somebody who's doing really well, or they're actually quite healthy, they just wanted to check in, you may find you just do this every year. And it's just one of those yearly tests that they do. So again, it depends on the person. Let's see. My patient has a lot of gut issues. Should I supplement while treating their gut, or wait and treat their gut first? Well, I'll be honest. I tend to wait and treat the gut first. Just like I said, you are what you assimilate, not you are what you eat. And so if you're giving people oral B6 and oral B12 and oral 5-HTP, and they have a lot of stomach intestine issues, then that it may not get absorbed. You may just get really expensive urine, which is funny since we're a urine test, but or expensive stool if it comes out, depending which way it goes out. So I tend to treat the gut first and then, supplement after the fact. You can consider other options: IV therapy, injections, topical, sublingual, dissolvable, and maybe go that route instead, doing B12 injections or sublingual B12 or magnesium topically. So you do get the benefits and you're treating their gut. Let's see. My patient has anxiety, so they are taking things for anti-anxiety that affect GABA. Will this also affect the test? I don't see that you are testing GABA. Keep in mind, anything that you do anti-anxiety inevitably affects the HPA axis. So if they're taking Xanax for anxiety, or they're taking something else calming, even pregnenolone, progesterone, L-theanine, GABA, phenibut, those things that can affect, they're calming to the whole system. So you may find their HPA axis was high, And now on those anti-anxiety things, it improves. So I don't see your testing GABA, not in these 6, not in these sticks. We are not, but stay tuned. The year is young. Can my patients do the test when they are sick? The flu is going around, but I just want to get the test done. We do get this question over and over and over again. We do not recommend testing when you're sick or when you have an active infection, especially a urinary tract infection, any kind of infection where somebody's on antibiotics, any kind of infection that maybe they're on a short course of steroids. They got a bee sting and, they're allergic to bees and it's right at the right part of their cycle. But or if they're on a detox, that's the other one. Yes, thank you, Mark. Just whispered that to me. So if they're doing any kind of like detoxification program, we don't tend to recommend it. So if they're sick, don't do it. If they're on antibiotics, don't do it. If they're on a prednisone pack for something acute, don't do it. If it's out of the blue, like, oh man, I woke up with, a urinary tract infection, but today's day 19. I'm like, oh, I know. I'm sorry. I wouldn't recommend it because infection and inflammation affect the test. They affect your hormones. It'll affect any test, any of them, your thyroid, your iron, no matter what, your stool. So no, get better from the flu, and then go from there.